Assalamu alaikum. Now we will consider lipid metabolism. First of all, we will have a rapid review about the biochemistry of lipoproteins in human body. Lipids generally are ubiquitous in body tissues and could play important and vital role in different functions of the cell, which can be summarized in the following points. First, lipids act as hormones or hormone precursors, as these are derived from, for example, for steroidal hormones derived from cholesterol. And also it aids in digestion, for example, bile acids and salts, which are also derived from cholesterol. 3, providing source of metabolic fuels and energy storage, represented by triglyceride in adipose tissues. 4, acting as functional and structural components in cell membrane, for example, phospholipids. 5, forming insulation to allow nerve conduction or to prevent heat loss. It is important, found in the nerve tissue and also in the subcutaneous tissue to prevent heat loss from the body as insulator. The basic components of lipids in human body include first fatty acids, which are straight chain carbon compounds of varying length, could occur as short chain fatty acids, medium chain and long chain fatty acids. These could be saturated with no double bonds or could be monounsaturated or polyunsaturated fatty acids. These fatty acids could be either esterified with different types of alcohols to form esters. Most of the lipids in the body are chemically esters by esterification of these fatty acids with different types of alcohols. Example of these alcohols is the glycerol, which is trihydric alcohol to form triglycerides, or could occur as free fatty acids or non-esterified fatty acids. Generally, triglycerides are transported from intestine when these are absorbed from diet as exogenous lipids to various tissues, including the liver and adipose tissues as lipoproteins. After their hydrolysis, these would be taken up by the adipose tissues to be re-esterified and stored as triglycerides. This is the general formula of what we've spoken about, different types of lipids. These are fatty acids, monocarboxy organic acids, the cholesterol structure with this hydroxy group at position or the carbon 3 can form esters with fatty acids to form cholesterol esters. Also, this alcohol, glycerol trihydric, can form esters with 3 fatty acids to form triglycerides, or this glycerol can form esters with 2 fatty acids and a phosphate group and nitrogenous base to form phospholipids. The second important type of lipids in human body is cholesterol, which is a steroidal alcohol that's found exclusively in animals and present almost in all cells and body fluids. It is important because it is a precursor of several physiologically important steroids, including the bile acids and salts and the steroidal hormones. The rate-limiting step for its synthesis is the 3-hydroxy-3-methylglutaryl-CoA reductase, which is inhibited, as you know, by the statins. It's also controlled by the negative feedback controlled by the intracellular cholesterol concentration. In plasma, about 2/3 of the cholesterol is esterified with fatty acids to form cholesterol esters. The lipoproteins, because the lipids are relatively insoluble in aqueous media, therefore cannot be transported in body fluids. Therefore, it should be transported in in more water-soluble compounds by forming complexes, spherical soluble protein complexes, to enhance their transportation in the aqueous media of the plasma. Lipids can be derived from food, could be exogenous or synthesized in the body, endogenous. The water-soluble, which represent the polar group of the proteins, also the phospholipids and the free cholesterol, would face the outer most of the particles surrounding the inner, which represent the nonpolar core of the lipoprotein particles, which contains the triglycerides and cholesterol esters. Lipoproteins generally can be classified according to their density and inversely reflects their size. The greater the size of the lipoprotein, the less dense. Also, the greater the ratio of lipid to the protein would result in larger size and lower density. Lipoproteins generally are classified into 5 main types. The first 3 are triglyceride rich because of their large size and they scatter light and can give a plasma turbid appearance, lipemic form, if they have high concentrations. The 5 main types including first, the chylomicrons, which represent the largest and the least dense lipoproteins, transport mainly the exogenous lipids that's absorbed from the dietary lipids in the intestine. Second, the very low density lipoprotein VLDL transport the endogenous triglycerides synthesized by the liver to other cells. 3. Intermediate density lipoproteins, which are transient form and usually not detected in measuring the lipid profile. It is usually formed during the conversion of very low density lipoprotein to low density lipoprotein. The other 3 types are mostly cholesterol rich and characterized by being smaller in size, include the low density lipoproteins. These are usually derived from very low density lipoprotein and mostly composed of cholesterol and act to carry cholesterol from the liver to the peripheral tissues. And 5th type include the high density lipoproteins, which are the most dense lipoproteins and involved in the transport of cholesterol from cells back to the liver, which represent the reverse cholesterol transportation. These lipoproteins can be further divided according to their density as HDL2 and HDL3. This table, which summarizes the different types of lipoproteins, of course, as you see, these had been classified, as we said earlier, that triglyceride rich particles, chylomicrons, and very low density lipoproteins, in addition to intermediate density lipoprotein, not normally presented in normal assay because these are transient, and the LDL particles, the LDL particles and HDL particles, which are the cholesterol rich particles. As you know, as you see there, named by the density. Their density is determined by the ratio of their lipid content to their protein content. As the lipid content increase, the size will increase and the density will decrease. These are arranged according to their density. If we see this table, the source of these lipoprotein particles, chylomicrons, are found in the gut from the exogenous or the dietary lipids. mostly rich in triglycerides. VLDL also rich in triglycerides endogenously made by the liver, and the LDL derived from VLDL, and the HDL is related from those taken up either from gut or liver by reverse conversion from peripheral tissues to the liver bag. Their composition, if we compare their composition, as you see, as we said, that these particles, which are the least dense and larger size, determined by their mass to their size. Protein percentage only 1%, while the remainder is all of this, it is lipid. Mostly it is triglyceride. This is for kilomicron. Also, these particles, classification of them based on their density, because they were first separated based on ultracentrifugation, and as you know, the density is determined by the mass to the size ratio. Therefore, we got these fractions. At the same time, because these contain protein in their nature, these can be separated by other technique depend on their electrophoretic mobility. Because kilomicron has only small portion of protein, which represent about 1% of their mass, only these cannot move and they stay at the origin. The VLDL particles, which are triglyceride-rich particles, endogenous triglyceride, as you see, the protein is much greater than that in the kilomicron, with also the greatest type of lipids is triglyceride. This can move in an electrophoretic field, electrophoresis field, to form a band known as pre-beta band. While the low density lipoprotein particle, as you see, the protein part increased up to 20%, with cholesterol about 50%, and by electrophoresis form the band beta, while the HDL particles, and this the most dense particle, the protein part represent about 50%, and the remainder of the lipids, mostly cholesterol and phospholipids, this represent by electrophoresis as the alpha band. By electrophoresis, which depend on the protein moiety ratio to the mass of the particle. As the protein is increased, the charge will increase and therefore it will move greater. Thus, alpha band will move faster and more distance from the origin, and will be the most distant particle from that of the origin. Here represent the kilomicron. The protein part, which form the important part of the lipoproteins, occurs or named as apolipoprotein. The term apo represent the protein part of the lipoprotein particles, had been found to occur in several types, either A or B or C or E types. Each of these have a specific function. Besides act as a carrier for the protein, of the lipids in the plasma, also it acts as a receptor to, or a guide for these particles to bind to the specific receptors in different tissues. The most important types of these apolipoproteins, the ApoA1 for the HDL to bind to its specific receptor, and ApoB48 for the VLDL, and ApoB100 for LDL particles to bind to their specific receptors in the hepatocytes to enhance the uptake by the hepatocytes. Because fasting plasma concentration does not normally contain kilomicrons, the triglyceride will mostly reflect the very low density lipoprotein particles which represent the endogenous triglyceride. Very low density lipoproteins can be quantified by precipitation technique when we use the specific kits to precipitate the VLDL and the LDL particles, and then can be easily assay the cholesterol content by the subtraction. Although other kits now are available to directly measure the LDL particles without the need to precipitate other particles and then to measure the HDL. The common technique or method to estimate the LDL cholesterol is based on the Friedwald equation, which enables measuring plasma LDL cholesterol concentration to be calculated by measuring total cholesterol and then subtract the HDL cholesterol and the triglyceride divided by 2.2 or which represent the VLDL particles. When this equation applied when the expression of concentration in millimole per liter, and if we use the traditional units when the expression of their concentration by milligram per deciliter, this factor will be 5. Triglyceride divided by 5 will represent the VLDL particles, which will be subtracted from total cholesterol together with HDL to give us the estimate for LDL cholesterol. There has been recent interest in the subdivision of the LDL particles into the smaller and dense LDL particles, LDL2 and LDL3, which appears to be more atherogenic and more easily oxidized than the larger LDL particles, which are mostly or first to be recognized in diabetic patients, which explain their ability to have atherosclerosis than the non-diabetic subjects. Another lipoprotein had been recognized in addition to the previously mentioned 5 lipoproteins called lipoprotein A, represented as LPA small, has been also found, which has a similar lipid composition to that of LDL, but it has a higher protein content. One of its proteins is called apolipoprotein A, which show homology to plasminogen and may disturb fibrinolysis, thus evoking thrombotic tendency, and the plasma concentration of lipoprotein A normally is less than 30 mg per deciliter and considered to be an independent cardiovascular risk factor. The proteins associated with lipoproteins are called, as we said, apolipoproteins. The ApoA, mainly as ApoA1 and ApoA2, is the major group that's associated with HDL particles. The ApoB series, most important type, ApoB100, which is predominantly found with the LDL particles, and it is the ligand for the LDL receptors in hepatocytes. The low-density lipoprotein has one molecule of ApoB100 per particle. This table summarizes the types of apolipoproteins and their common and important functions. ApoA occurs in several subclasses, as I mentioned earlier. ApoA1, found in the chylomicrons and HDL, act as LCAT activator, lecithin cholesterol acyl transferase activator. ApoA2, also found in chylomicron and HDL, and act also as LCAT activator. ApoB48, mostly found and important for the metabolism of chylomicron and very low density lipoprotein. It is made important for their secretion. ApoB100, as we say, it is mainly found in the LDL, which is derived from VLDL and IDL, their precursor. By their important functions of the ApoB100 as a site for the LDL receptor binding in the hepatocytes to enhance the clearance of LDL from circulation. ApoC2, also found in chylomicron, HDL, VLDL, and IDL, it is important lipoprotein lipase activator. And ApoC3, it is the lipoprotein lipase inhibitor. And ApoE, it is found mostly in the intermediate density lipoprotein and also in the remnant particles for the identification by the specific receptor binding site. In cases of hyperlipidemia, the lipoprotein pattern has been classified according to Fredrickson classification based on the electrophoretic mobility, which is the type of classification identified by the WHO to study the dyslipidemia. 4 principal bonds had been recognized based on the relative position by the protein electrophoresis, either the alpha band, which represents the HDL, or the pre-beta, represent the VLDL, and the beta band represent the LDL, in addition to the chylomicron, as we said, occurs in the origin. Ultracentrifugation, which is the basic technique used to identify the particle density, or also we can use the electrophoretic technique. However, it is rarely used in routine clinical laboratory or only used for research. These may require complicated apparatus and experienced operators. Now, we will study the disorders associated with lipid metabolism. The study of hyperlipidemia, it is of considerable importance because its involvement in the cardiovascular diseases. Fredrickson had first to recognize or define the hyperlipidemias and classified into several types based on the electrophoretic pattern. However, the Fredrickson classification had put lipidology in the clinical map, and it was not a diagnostic classification. It just gave a little clue to the etiology of the disorder, and all the phenotypes of hyperlipidemia can be either primary or secondary. For Fredrickson classification, by this type, we consider the primary types of hyperlipidemia, which is mostly related to abnormalities or genetically determined abnormalities in lipid metabolism, whereas the secondary type is related to other metabolic abnormalities. This table found in your textbook by Kroc represent the Fredrickson classification, which is the system by which the WHO classify the hyperlipidemia or dyslipidemia as these 5 major classes. Type 1, characterized by increased chylomicron on electrophoresis, or type 2A, which increased beta lipoprotein الباند اللي هي تمثل لنا ال LDL, and type 2B increased beta and pre-beta lipoprotein اللي هي تقابل لنا ال LDL and VLDL, and type 3 broad beta اللي هي هالحالة حتظهر لنا ال abnormal ال IDL particles, و type 4 increased pre-beta اللي هي تمثل لنا endogenous triglyceride particle VLDL، والتايب 5 increased chylomicron and pre-beta اللي هي حسر عندنا زيادة بالtriglycerides both الchylomicron and VLDL. Now more realistic classification for studying. This lipidemia is based exactly for the primary hyperlipidemia has been made as follows. First, the chylomicron syndrome. This can be related to familial deficiency of the specific enzyme lipoprotein lipase. This defect is related to an autosomal recessive disorder which usually affects 1 in 1 million people. The gene for lipoprotein lipase is found on chromosome 8, and genetic studies have shown insertion or deletion in this gene location. Lipoprotein lipase generally is involved in the exogenous lipoprotein pathway by hydrolyzing the chylomicron, as we said earlier, represent the exogenous triglyceride to form chylomicron remnant, and also in the endogenous pathway to convert VLDL to IDL particles. This chylomicron syndrome is usually presented during childhood with abdominal pain, which is related to acute pancreatitis. Also, there is probably no increased risk for coronary artery disease, but gross elevation in plasma triglyceride due to the accumulation of the uncleared chylomicron in lipoprotein lipase act to hydrolyze the triglyceride component of these chylomicron particles, and if this enzyme is deficient, these particles will remain in circulation and accumulated and build up. The lipid stigma picture العامة اللي تظهر عند those patients include eruptive xanthomata اللي هي تكون itchy xanthomas اللي هي accumulations of lipids under the skin, and hepatosplenomegaly in addition to lipemia retinalis. This picture represents the lipemia retinalis in patients with lipoprotein lipase deficiency and chylomicron syndrome. Later on, they found there is another variant for this syndrome, chylomicron syndrome, include the circulating inhibitor of lipoprotein lipase, and not only deficiency of the lipoprotein lipase enzyme, but also presence of inhibitors of lipoprotein lipase and also deficiency of its activator ApoC2, not only the enzyme. ApoC2 deficiency is also inherited as an autosomal recessive condition which affects about 1 in million people. And the gene responsible for this ApoC2 deficiency is located on chromosome 19, and mutations results in low plasma concentrations had been detected. This will be left to the second lecture. Thank you.